viruses invade cells. The two main types of nucleic acid are the DNA and RNA. Both DNA and RNA are made from nucleotides, each containing a 5-carbon sugar backbone, a phosphate group, and a nitrogen base. DNA provides the code for the cell's activities, while RNA converts that code into proteins to carry out cellular function.
A nucleotide is a basic building block of nucleic acid. A nucleotide consists of a sugar molecule, either ribose in RNA or deoxyribose in DNA, attached to a phosphate group and a nitrogen-containing base. The bases used in DNA are adenine for A, cytosine for C, guanine for G, and thymine for T.
Viruses are tiny particles, smaller than prokaryotic cells. They are a strand of genetic material within a protective protein code called a capsid and cannot replicate without help. But they have nucleic acid and can evolve. The virus spreads through the air and droplets. When the virus enters the cell body, releasing RNA, then the virus RNA invades the cell nucleus and takes over. The viral RNA uses the host cell to create new RNA, acts as molecular message instructing our host as machinery to read the template and translate it into protein that make new viruses particle and assemble more viral particle. The hijacking persists as the human host cell continues to generate more copies of the virus assembles. These copies into viral particle to tropic density to the edges of the cell for release. Each infected cell may produce or release millions of copies of the virus, which can then go on to infect other neighboring cells.
So the effect of virus variants on COVID-19 vaccine. All viruses, including SARS-CoV-2, the virus that causes COVID-19, evolve over time. When a virus replicates or make a copy of itself, it sometimes changes a little bit, which is normal for a virus. These changes are called mutation. So a virus with one or more new mutation is referred to as a variant of the original virus. So what causes a virus to change to a new variant? So when a virus is widely circulating in a population and causing many infections, the likelihood of the virus mutating is increasing. So the more the opportunities a virus has to spread, the more it replicates, and the more opportunities it has to undergo changes. So most of the vital mutations have little to no impact on the virus's ability to cause infection and diseases. But depending on where the changes are located in the virus's genetic materials, they may affect the virus's properties, such as the transmission, or for example, it will spread more or less easily or severely. It may cause more or less severe diseases.
Variants of the COVID-19 virus have on vaccine. The COVID-19 that currently in development or have been approved are expected to provide at least some protections against the new virus variants because these vaccines elicit a broad immune response involving a range of antibodies and so. Therefore, changes or mutations in the virus should not make vaccine completely ineffective. In the event that any of these vaccines prove be less effective against one or more variants, it will be possible to change the composition or the vaccine to protect against this variant. How can we prevent the future the new variant of COVID-19 virus? Stopping spreading at the source of remain key. Current measure to reduce the transmission including frequent hand washing, wear masks, distancing and good ventilation, and avoiding crowded places. Closed settings continue to work against new variant by reducing the amount of the viral transmission and therefore also reducing opportunities for the virus to mutate.
Why is it important to get vaccinated even if there are new variants of the virus? Vaccines are a critical tool in the battle against COVID-19, and there are clear public health and life-saving benefits to using the tools we already have. We must not put off getting vaccinated because of our concerns about new variants. And we must proceed with vaccination even if the vaccines may be somewhat less effective against some of the COVID-19 variants. We need to use the tools we have in hand even while we continue to improve those tools. Although vaccines afford very high protection, infection with the Delta and other variants remain possible. Fortunately, vaccination, even among those who acquire infections, appears to prevent serious illness, hospitalization, and death from COVID-19. We are all safe only if everyone is safe.
Providing it as a
Coronaviruses invade cells through so-called spike proteins. The spike protein is the major surface protein that binds to a receptor, which acts like a doorway into a human cell. Once the spike protein and the surface of the human cell membrane fuse together, the genome of the virus is able to enter the human cell and begin infection. Understanding the shape of the spike protein in COVID-19 is the key to figuring out how to target the virus and accelerate potential vaccines. Jason.
RNA vaccines and conventional vaccines. The manufacturing process for RNA vaccines is successfully developed and approved, may speed up the response to infectious disease outbreaks. So what's the difference between RNA vaccines from conventional vaccines? In production, the conventional vaccines, most vaccines against viral diseases are made from viruses grown in chicken eggs or mammalian cells. The process of collecting the viruses, adapting them to grow in the lab, and shipping them around the world can take months and is complex. For newly emerging viruses like SARS, coronavirus, for which a new vaccine is needed as quickly as possible, these steps may slow down development. While RNA vaccines, which encodes an antigen of the infectious agent made from a DNA template in the lab, the DNA can be synthesized from an electronic sequence that can be sent across the world in an instant by computer. Currently, it takes about a week to generate an experimental batch of an RNA vaccine. In biosafety, the conventional vaccines growing large quantities of virus to make each batch of vaccines creates potential hazards. While in RNA vaccines, no virus is needed to make a batch. Only small quantities of virus are used for gene sequencing and vaccine testing. In immune response, the conventional vaccines, the antigen, or a piece of the virus is injected into the human body. Upon recognizing the antigen, the immune system produces specific antibodies in preparation for the next time the body encounters the pathogen. While RNA vaccines, the RNA is injected into the body and enters cells, where it provides instructions to produce antigens. Then the cell presents the antigen to the immune system, prompting T-cell and antibody responses that can fight the disease. Next is the flexibility. In conventional vaccines, each new vaccine requires a bespoke production process, including complex purification and testing. And RNA vaccines, we anticipate that the production process for RNA vaccines may be able to be scaled and standardized, potentially enabling replacement of the sequence encoding the target proteins of interest for a new vaccine with minimal changes to the vaccine production process.
Good day everyone, so my name is Rosalie Andayawan. Emergency uses of the Pfizer Biotech COVID-19 vaccine have not been approved or licensed by the FDA, but have been authorized by the FDA under an emergency use authorization to prevent coronavirus disease 2018. In individual 5 years of age and older, Comirnaty is licensed by the FDA for individuals 16 years of age and older. In addition, Comirnaty is under EUA for individuals aged 12 to 15 years of age for their third dose for certain individuals 18 years of age and older. The emergency uses are only authorized for durations of the declaration. The circumstances exist justifying the authorizations of emergency use of the medical product under Section 564 of the FD&C Act unless the declaration is terminated or authorization revoked sooner. Thank you.